We are honored to welcome Dr. Ramon Parsons, director of the Tisch Cancer Institute here at Mount Sinai. Dr. Parsons received his MD and PhD from the State University of New York at Stony Brook. He continued his education at Johns Hopkins University School of Medicine as a postdoctoral fellow, where he and his colleagues discovered that inactivation of DNA mismatch repair genes causes hereditary colorectal cancer. He has become a leader in establishing the importance of P10 and the PI3K pathways. He received the 2011 American Association for Cancer Research Outstanding Investigator Award for Breast Cancer Research and has been inducted into the Johns Hopkins University's Society of Scholars. In 2013, Dr. Parsons joined the faculty at the Icahn School of Medicine at Mount Sinai as Ward Coleman Professor in Cancer Research, Chairman of the Department of Oncological Sciences, and co-leader of the Cancer Mechanisms Program at the Tisch Cancer Institute. Most recently, Dr. Parsons was appointed Director of the Tisch Cancer Institute at the Icahn School of Medicine at Mount Sinai and Director of Mount Sinai Cancer in the Mount Sinai Health System. Please join me in welcoming Dr. Parsons.
Thank you for that very kind introduction. And I guess I'll get started with the talk. So these are my disclosures. So this is a slide from Gady Getz's lab at the Broad that was published in 2014. I don't know if you caught this paper or not. Looks like you did or you didn't. Anyway, what this is a blow up of the significance. Let's see, how do I? Oh, right. So this is just a blow up of a very important figure where they looked at 21 different types of cancer across almost 5,000 types. 20 different cancer types, over 4,700 tumors, and these are basically Q-value quantitations on the Y and the X-axis. This is within a specific cancer type, and this is across cancer types, so things that may not be that common with any one type of cancer, but where the cumulative evidence in 21 different cancer types starts to emerge as significant. So you can see this gene here, ARID2, here in this section here, turned out to be quite significant across cancer types, but relatively low frequency, but not strong within any one cancer type. And then over here, you have something like TET2 and WT1, Wilms tumor suppressor, that are very specific to certain kinds of cancers. And then up on the right-hand corner are the ones that are most significant in many different cancers and within and very common within specific cancers. And so the third most common gene, and what was nice about this study is it used one set of standard for analyzing the genomic data. And when they did that, P53, TP53 was the most significant tumor suppressor across cancer. The second most significant was PIK3CA, which encodes for PI3 kinase alpha. And the third most significant was P10, which I'll be discussing. And then the fourth was KRAS, BRAF. And then it's hard for you guys to see all these different gene names. But the point is that when one does an unbiased analysis of the genome, P10 is really commonly altered in a lot of different kinds of cancers. So, and we know that, by the way, these genes like P53, PIK3CA, KRAS, BRAF, P10 are drivers. We know from our work for the last several decades that these genes actually cause cancer. They turn on signaling pathways. They turn on metabolic pathways that drive tumor growth or block tumor death. And so, and the other thing that I should point out is these are from primary tumor biopsies, not metastatic tumors, and that these are in the context of all the other stroma. One can see these mutations occurring in the tumor. So The P10 tumor suppressor, for those of you that aren't familiar with it, is a phosphatase. That's how it got its name, P for phosphatase. And tensin, it's a homolog of another gene called tensin, or protein called tensin, that has homology to P10, and it's on chromosome 10. And in addition to being mutated somatically, so that prior slide was data of somatic mutation, not germline mutation, in cancer samples. We can see germline mutations in P10 that are associated with Cowden's disease and hamartoma tumor syndromes, autism, and macrocephaly. And the phosphatase that P10 encodes antagonizes the PI3 kinase pathway. So, once we understood this relationship, we started to study the PI3 kinase pathway in depth to see if it also was mutated in cancer, and I'll talk about that later. So, okay, so loss of P10, as I sort of alluded to before, is a driver. It can cause many different oncophenotypes in the cell, and it can be inactivated or inhibited through multiple different mechanisms. Actually, probably in certain kinds of cancer, mutations are not very common. They're basically, there's epigenetic or other protein-based mechanisms of like inhibition that could be regulating P10 and downregulating. And on the bottom is just a little cartoon of the domain structure. We have a phosphatase domain, a C2 domain, a tail domain, and a PDZ binding domain, which have different roles in the cell. And in terms of where this P10 can work, it can work inside the nucleus, but the action where most of what we know about is on in the cytoplasm and on the plasma membrane, where it regulates the PI3 kinase signal downstream of receptor tyrosine kinases. It also can get outside of the cell and circulate around and come back in. And this was revealed relatively recently when we found another, and others have found other isoforms of P10 that are longer, that have additional domains. These are made at much lower levels than regular P10 because they use leucine initiation or other alternative translation initiation start sites, so they're very low abundance, but appear to have a physiologic function that is being studied as an ongoing effort. And, so anyway, trying to understand P10 as a molecular genomic entity came out of the work from the Bigner lab at Duke, where they showed in 1984 using karyotyping of glioblastoma that chromosome 10 was, one copy was often lost when karyotypes were done on samples grown from patients' brain tumors. And 1984 was also the year that RB was discovered as a tumor suppressor. So the hypothesis was there that there was potentially a tumor suppressor located on chromosome 10. And we, years later, basically jumped on this bandwagon in my lab at Columbia to try to identify this tumor suppressor, and we were aided by a lot of different mapping work that was going on in the genome at that time. But basically, the bottom line is, if you look here, we did analysis of a series of different, we call them PDXs now, patient-derived xenografts. We used to just call them xenografts back then. But we had a large series of breast tumor xenografts that we got from Texas at the Stellan Institute. And they sent them to us, and we were able to make DNA from them. And two of them were missing a marker in this locus here. And that led to us identifying the P10 gene because it was just spanning the deletions that were where DNA was missing from the tumors. And we just did something called positional cloning to identify a transcript, which is shown here, present in every cell line we could see, except for something that was deleted or mutated. And this is just the domains, the homology between P10 and phosphatases and tensin. So here's tensin and P10. Here's P10, this is the yeast version of P10. This is CDC14. And these are members of different members of the tyrosine dual specificity phosphatase family members. This was the first deletion that we identified in P10. And here you can see here are some of the first mutations using actually glioblastoma samples from Columbia Hospital, where we were seeing mutations in the, here's an example where this bubble is. A band is missing right next to the bubble, and you can see there's a mutation here in this case that's loaded right next to it. So these are two cases, case 5 and 6 loaded side by side, where there's a mutation in 6. There's other examples in here too. You can see where, and you can we were able to map these to the open reading frame or to splice sites, and they led to inactivating mutations. When we replaced P10 into breast cells, we decided to focus on breast cancer because when we sequenced about 20 breast cancer cell lines, we found 5 of them were mutated. And here you can see Nexin 5 here is a marker of apoptosis is induced when we add P10 into these into one example of a P10 null cell line, and that we could rescue it partially with a caspase inhibitor. And this was seen in several of the different tumor cell lines we were studying. Other cell lines didn't respond this way. They responded with more of a cell cycle arrest phenotype. And another area that we saw where early on was working with Ken Yamada at NIH at the time. There's no need to actually go look at the details of this data other than to say that we could see using sense and antisense molecules and also an inducible system that P10 could inhibit cell migration. So here was a very important paper from Jack Dixon's group published in 1998, the year after we identified P10, that determined that P10 was on the PI3 kinase pathway, where they showed that P10 was a lipid phosphatase that could dephosphorylate the third position of PI345 P3, or phosphatidylinositol 345 trisphosphate. And you can see here in this cartoon, and in the study, they showed that P10 could inhibit the signal from insulin to generation of PIP3. And around that same time, it was just been reported that from Luke Cantley's lab that AKT could bind to PIP3 and activate downstream signaling. So, and this was their model, which they didn't actually test the relationship between PIP3, P10, and AKT in this paper, but like we and others did this immediately and published that we could see that P10 was downregulating AKT phosphorylation, and also data shown here. This is this is basically looking at GFP positive cells, or viable cells. We could inhibit viable cell with P10 GFP, and that we could rescue this with either BCL2 or myristylated AKT. We also, and others did the same, found that when we add P10 to cells, we could lower the level of PIP3 in the cell. This is in a P10 null cell setting. And then in parallel, we knocked out P10 in mice and wanted to look at whether or not that had led to tumor development, as was seen in humans. And so there was a very good mouse homologue. The open reading frame for the regular P10 was very conserved. It contained all the same amino acids except for one, which was a serine to a threonine. And the mice, the heterozygous mice, developed many different kinds of tumors. This is just an example of some lymphadenopathy that's very common in the mice when they reach, like, you start to see it starting around 6 months, or before that even. these are the different kinds of tumors they developed. And one of the really satisfying things that we did was that, at least in many of the tumors, we could see very strong evidence of AKT activation actually in the tumors or lesions. These are really pin lesions. They're early changes in the prostate are shown here, and you can see this hyperplasia and dysplasia in a setting of the normal epithelium shown here. And there's lower P10 staining in these lesions, and you can also see activation of AKT on the plasma membrane shown here. Another feature that we could see in lesions of the prostate and breast and other tumor types, pheochromocytomas, was increased proliferation in the tumors. And based on that, we wanted to look at whether or not we could target the signaling pathway with a drug in the mice to determine whether it would have a therapeutic benefit. And so at the time when we were doing this back in like 2000, we didn't have this detailed knowledge of the signaling pathway where PI3 kinase activates PIP3, which can activate AKT, which then can further regulate through several levels of regulation, the mTOR TORC1 complex, and we could target. But we did know that simply that PI3 kinase could regulate the TORC1 activation in S6 kinase. The details here hadn't been discovered yet. But basically, we had this sort of indirect knowledge. We knew PI3 kinase could activate TORC in S6 kinase, and we knew that S6 kinase was downstream of TORC1. And so we decided to inhibit with Rapalog that we were able to obtain from Wyeth. And then we could see that, that we could lower S6 kinase activity and also could block tumor progression in our mouse model. This was relatively straightforward to do in mice, but obviously it's been a long road to try to figure out how to target this pathway in cancer effectively without toxicity. But further, just to further discuss this a little bit, we know that when you get rid of P10, and I think I showed, you can see this in this prior slides, but basically you upregulate the level of PIP3 on the plasma membrane, and this unleashes this signaling pathway in the cell that increases proliferation, actually inhibits cell death, and activates various metabolic pathways in the cell. In terms of clinical kinase inhibitors that are in clinical trials or approved, we have inhibitors for PI3 kinase and AKT that are undergoing clinical development now, and also there are approved mTOR inhibitors out there that people have tried. And we know that an mTOR inhibitor can be used in combination with hormone therapy for breast cancer and metastatic disease. It's quite toxic, but that is out there. And then we also know that many of the PI3 kinase inhibitors have been quite dirty, as have the AKT inhibitors been, and they have quite a bit of toxicity. But pharmaceutical companies are working on improving these profiles by making actually better drugs that are more specific. One thing that came out last year I was very excited about, came out a clinical trial in metastatic prostate cancer was actually found. First, you don't have to read this whole thing, but this is the first clinical demonstration supporting P10 loss as a predictive response biomarker for metastatic castration-resistant prostate cancer. So this was an AKT inhibitor that, in combination with abiraterone, they were seeing efficacy in this population of men. So hopefully, and one of the nice features of this drug is, unlike prior versions of AKT inhibitors, it shows some genotype specificity, meaning if there's like a P10 mutation, it tends to be more effective than in a P10 wild type tumor. So anyway, so I'm going to, those are sort of my overview slides, and I'm going to now go into some of the recently published work that came out of the group. This work, in this case, came out earlier this year, and it focused on using mouse embryonic fibroblasts, which we can, that are floxed for P10, so we can use Cre to delete P10, or use adenovirus that doesn't contain Cre as a control. And when we do this, we see increased proliferation in the knockout cells, increased BRDU, focus formation, and we also found out that if we remove glutamine from the media, that we could basically, this separation you can see here, we could shrink to here, and that showed us that maybe there was something going on with the metabolism of glutamine in the cells. So we did some glutamine labeling using stable isotopes for glutamine, and did flux analysis and steady state metabolomics. And just, this is a lot of data, and I don't expect you to be able to take a look at. You're welcome to look at the paper, but basically when we did a variety of different analyses, we could see that nucleotide metabolism, and particularly pyrimidine metabolism, was upregulated both at the steady state and flux levels in the cells that are P10 null. And so when we looked at Stryer, at this pathway that regulates the production of pyrimidines, and OMP is a pyrimidine precursor, we saw that dihydroorotate dehydrogenase, shown here, was a critical component of this de novo synthesis pathway. So we decided to take a look at this in further detail. And so what we did was, you can see here that when we looked at this, this is a drug A771726 that inhibits DHODH, and we could see basically when we, and I'll just point to this lower right-hand corner, that when we, in the knockout treated cells, we could see evidence of replication forks co-localizing with DNA damage foci very, very nicely in confocal imaging, showing that we think the DNA damage that we could see when we inhibited this occurred as a result of at replication forks. And also when we treated cells with a DHODH inhibitor, we also saw evidence of chromosome breaks, and this is after a few days in the presence of the drug, allowing for cell cycles to try to occur, and you see pulverized chromosomes in P10 null cells. Now, in this case, this is wild type and null P10 cell lines that we made in the lab from mouse breast lines that have either P10 knocked out plus MYC, or MYC plus PI3 kinase mutation as a control. And then lastly, for the important for us was to try to understand, like, why were we seeing this DNA damage, and we looked into this, and we could see that, and it had been reported previously that top BP1, which is critical for activating ATR checkpoint activation, was upregulated in its phosphorylation was increased in P10 null cells, and we expected that because it had been reported that this phosphorylation site is regulated by AKT, and since P10 loss is associated with high AKT, it's not surprising. But basically, and we also asked whether we could see any evidence of co-localization of top BP1 and DNA replication, and we could normally, but when we lost P10 we couldn't. But really, the most important experiments are on the bottom here, where we transfected mutants of TOPP1 and CHEK1, which is downstream on the ATR signaling pathway, that can't be, we made mutations in sites that are AKT phosphorylated, and we made alanine substitutions at these sites, and we could see evidence of reducing the DNA damage in the presence of a DH or DH inhibitor, leflunomide, or over here, we could actually just rescue some of the cell death that we were inducing with the drug. So as I alluded to at the very beginning of the talk, PIK3CA, which includes PI3 kinase, alpha, and P10 are very commonly altered. RAS actually is also very commonly altered. It's known to directly interface and activate PI3 kinase alpha. We also know, although they're not as commonly mutated, AKT1 and AKT2 are mutated in different kinds of cancer, and PDK1, which is required for phosphorylating AKT on one of the key sites for its regulation, is amplified in a lot of cancers, particularly breast cancer. And finally, solving a long-standing mystery about what the second AKT kinase was. A few years ago, it was discovered at Harvard that TORC2, which is the second major subunit class of mTOR kinases, can be PIP3 regulated and through this then directly phosphorylate AKT at its second site, which then, so in terms of mutations, we see mutations in P10, amplification of PDK1, mutation of AKT. PREX, which I'll talk to about later, can also be mutated and overexpressed and amplified, and PI3 kinase alpha and RAS are mutated. And obviously, tyrosine kinase receptors can be altered as well. Just to talk a little bit about PI3 kinase, because it's so frequently mutated, we identified that it was mutated very commonly in ER-positive breast cancer in 2005, and showed that, had been previously reported by the Vogelstein group in colon cancer, that these mutations occurred in hotspots, one in the kinase domain and one here in the middle of the protein. Both of these, subsequently, we know, lead to activation of signal transduction and they can transform cells and cause tumors in their own right. Relatively weak, though, when knocked in at the endogenous level, but when one combines them with another oncogene or tumor suppressor mutation, they're very cooperative and lead to tumor development. Another thing that I think just want to point out is when you look at breast cancer using, this is back in 2007 when we were using microarray analysis, we were able to identify a gene expression signature associated with loss of P10, here's signature present. It's about a 250 gene expression signature, and we could see its association with a variety of other markers and genetic changes that were common. This led us to identify that P10 is lost very commonly in triple-negative breast cancer. And the other key thing that is really important to understand about the PI3 kinase P10 pathway, and part of the reason why it's so difficult to study therapeutically or to actually use it as a therapy target, is that it is, because it's so involved in the regulation of signal transduction and metabolism and growth and mitogenesis, it's subject to a lot of different feedback loops, which basically, whenever you try to inhibit it, the cell tries to restore anything you're trying to inhibit. So it's subject to critical homeostasis. And just to make an illustration of this, when you restore P10 in cells, you see feedback activation of AKT, PI3 kinase, IRS. These are adapter proteins that regulate, and also receptors. And these can be at the transcription level, they can be at the protein half-life level, they can be at the post-translational modification level. So it's really pretty much in any way you try to inhibit the pathway, there is there's evidence that the cells try to find a way. This often takes time. It takes as long as, in some cases, like 24 hours to maybe 2 or 3 days for this adaptation to occur, but it does happen. So another big sort of hurdle for trying to target this pathway in cancer has been that we've also identified, in addition to feedback, several mechanisms of resistance to PI3 kinase inhibitors. These are basically come from genetic studies done in laboratories where one is either screening for or is identifying de novo resistance, either in the presence or absence of an inhibitor. But the basic point is there's many different ways a cell can bypass an inhibitor of PI3 kinase in the cell, one of them being MYC, also receptor tyrosine kinases, RAS. And since these are such common players in cancer, these are a real issue if one wants to use this as a target for therapy. So one idea we had in using a MYC model with PI3 kinase or P10 mutation was to, where we saw very similar feedback. Basically, here's a cartoon showing we're inhibiting PI3 kinase with a drug, and this is leading to a change from pre-treatment to treatment in the level of activity of the receptor tyrosine kinases that are being turned on from the gray to the black arrow at the transcriptional level. And so this is a component of the feedback, but we're able to address that using a BET inhibitor that targets BRD4 that blocks at least the transcriptional component of the feedback and leads to a more efficacious inhibition of the pathway. Now, of course, that could come at some kind of toxic price, but at least we were able in this setting to find a way to do this. And my hope is that in this case, this inhibitor, while very effective, is a relatively dirty inhibitor in the sense that it hits multiple kinases and not just PI3 kinase alpha. So, okay, so in addition to, just a couple of other things to point out is to talk a little bit about PREX2. So the PREXs, there's PREX1 and PREX2, are RAC-GEFs, and PREX2 binds, well, we first got into understanding a little bit about PREX2 through identifying that it binds to P10, and that PREX2 is able to inhibit P10 activity. And here, we also know, and so that's sort of one way it can work. Another way it can work is it's actually can bind to PIP3 just like AKT or PDK1 can. And in this case, once PREX binds to PIP3, it activates its guanine nucleotide exchange activity, which then can load GTP onto RAC. Now, interestingly, RAC can activate PI3 kinase beta, and so you could get into this feed-forward loop in a setting where there's high levels of PIP3 and excess PREX1 or 2. Understanding the role that this plays in the physiology of cancer is still unclear, but I can tell you it's very exciting in the sense that recent studies have now shown that RAC actually is a critical mediator in activating glycolysis in tumor cells. And this is work from Gerburg Wolf's lab at Harvard. And okay, so this is just some of the early data where we showed that P10 could be inhibited by PREX2 after finding that they could interact. And more recently, in a couple of papers we published in JBC, we've determined that PREX2 can basically be phosphorylated after activating insulin or GPCR signaling. It can activate PAC, which then leads to a phosphorylation of PREX2 on many, many different sites. And then this leads to a dissociation of PREX2 from the membrane. And then we've shown that phosphatases can dephosphorylate, and presumably, though we don't have evidence for this, that this could be a cycle that could be repeat the ability of PREX2 and RAC to signal on the cell, and that the reason for this feedback loop is simply to block the permanent activation of RAC in the cell and have a clock on the signal. And we also know that P10 is continuously associated on this cycle, so it's also possible that P10 could go along for the ride and then be displaced from the membrane and then come around. And this could also be a possible way for P10 to be inactivated through just localization at the plasma membrane rather than direct inhibition. And then last on this topic, I just wanted to mention that we know that P10 can also inhibit PREX2's GEF activity towards RAC. And we recently showed now in 2015 that the original study that showed that P10 could inhibit migration appears to occur through the ability to inhibit RAC activity through PREX2. Okay, so the last part of my talk is about a paper, is going to go focus on transcriptional downregulation of P10 in breast cancer. We know that promoter methylation and other chromatin modifications, LOH, altered transcription factors, MIRS, can lead to downregulation of P10 in different kinds of cancer. But specifically in breast cancer, there's really no good evidence that I'm aware of that MIRS are playing a role here. So we've been focused more on the promoter and enhancer landscape here being downregulated because we clearly see when we see loss of P10 mRNA and protein, in many cases it's definitely not associated with a genetic alteration in the gene itself. And I'm just going to bring up this concept of haploinsufficiency of tumor suppressors, which I'll get into in a little bit more detail because it's pertinent here. But basically, when you have the classic 2-hit model, a heterozygous cell has no phenotype. But in a haploinsufficient tumor suppressor model, one copy loss of a tumor suppressor can lead to a phenotype. Often, this may require another gene to be altered in combination or in cooperation with this gene dose effect. But in this case, we know that this type of heterozygous or haploinsufficient can occur for P10 and other tumor suppressors as well. And so when we were exploring the relationship between P10 and other changes, we tripped upon an association between P10 expression loss and P53 mutation, which is shown here. There's a P10 mRNA is downregulated in green, and the P10 protein is tending to be downregulated, and the P53 mutation is also in green, is showing that these are clustering, tending to co-associate, and we could also confirm this P53 mutation relationship to P10 in our own data by just QRT-PCR of the same samples that we'd done an array analysis on. And also, we could see this relationship in large data sets that are published in public. So based on this, we asked the question, could P53 regulate P10 and other important targets at baseline? And the background of this is that most work in the field of P53 considers P53 to be a stress-activated gene that does its job in response to extracellular or intracellular stress. And so what we're saying would have to be occurring in the body, where P53 would be regulating P10 under, I guess, physiologic stress potentially, not sort of environmental stress. So that's a bit of, I don't know, a bit complex. But in any event, most thought is considered that it's these like DNA damage events that are involved in tumor suppression. And our data would sort of argue if we could see something that it might be, there may be something more sort of physiologic in terms of the in vivo tumor suppression may be through some level P10 expression, a level regulation that just occurs tonically in the body through P53. But anyway, P53 mutations cluster in the DNA binding domain. They block the ability to activate a bunch of different transcription targets. One other thing that's in the literature that's really interesting and mysterious is that people have made different mutations, different groups have made mutations in lysine sites that are key for the regulation of P53 and its ability to activate some genes but not others. They don't disrupt DNA binding for P53. And what's interesting is that if you get rid of 3 of these lysine sites shown here, you can see that tumors don't develop as well as if you just get rid of the whole P53 protein completely. And what's also interesting is that when you make these mutations, a lot of these stress-activated genes are no longer activated in the mice. So it really argues that maybe sort of this dogma or model of how P53 suppresses tumors needs to be modified. Okay. So I'm going to skip through this slide because it's more just of the same thing. But basically to say that P53 is expressed at measurable levels in the body across a variety of tissue types. And so in general, what we decided to do, just to cut to the chase, because I'll run out of time, is to see if we could see basally express P53 in a human cell line that's wild type for P53 called MCF10A. It's a human immortal line that is not derived from a tumor, and it can be easily grown in culture. And we decided to do ChIP-seq on using an antibody to P53 that many people have been using already, and to determine if P53 could be binding to P10. And then we looked at publicly available data for another cell line that has wild-type P53 that's been a model for a P53 function, U2OS. And we could see just at baseline, in fact, in our data and also public data, without adding any DNA damage or nutlin or any other activator, that we could see baseline signals where you'd expect them at P21, MDM2, and MIR34A, which have been reported to actually have baseline binding of P53 in the past. And then we looked at P10, and we could see nicely a very nice enrichment for a signal in P10. But we also, what was surprising was we could see these sort of baseline binding in other tumor suppressors, like STK11, which is a major tumor suppressor in lung cancer, and UTX, which is mutated in a variety of different kinds of cancer at relatively high frequency. And then we asked some other very straightforward questions like, could we see if we got rid of P53 by RNAi, could we interrupt the binding? And we could. We also asked if we just took uncultured cells like PBMCs, would we see binding of P10 in these different genes? And we could see that as well. And then we just rank ordered our ChIP-seq data and looked for, are there any other tumor suppressors in there? And we could see that about a dozen different tumor suppressors were at the leading edge of our ChIP-seq for P53 in these cells and also in the U2OS cells. And then we just said, okay, what's an unbiased way to look at this? So we took, there's a database called COSMIC for catalog of somatic mutations in cancer at the Sanger Center in Cambridge, and they have a list of oncogenes and tumor suppressors. And we asked whether this was, the list of tumor suppressors was enriched on our list of P53 targets, and it was, while a list of oncogenes was not, even though P53 binds to oncogenes like MDM2. And so suggesting that this was more than just a fancy on our part. And we also could see, when we look at these tumor suppressors, we define them in the literature based on whether they could cause, this is just our criteria, but basically almost all of them have a genetically engineered mouse model of cancer that's associated with neoplasia. When we looked at the peaks, they actually contained conserved P53 binding sites, so that when these peaks were not just, they actually had very, very strong binding sites for P53 that, which, this is an area that has been under investigation since the 1990s. So that was quite comforting. And then the other thing, we looked at publicly available data from cell lines where P53 ChIP-seq had been done, and we categorized them based on whether they had P53 mutation, wild-type cancers, wild-type P53 in cancers, or wild-type P53 in non-transformed cells, like the MCF10A we started with, here IMR90, or human foreskin keratinocytes. And you can see, even though we don't see all the same peaks, we tend to clearly see many of the same peaks. In the wild-type lines for P53 that are cancers, we see some cancers that have very low numbers of peaks. And interestingly, MCF7 has a MDM2 hyperactivation, and this leads to low levels of baseline P53 activity, which makes sense. But particularly notable are the P53 mutation cell lines, which didn't have any peaks at all, and these are in the DNA binding site, suggesting that when mutations occur in P53, these genes are no longer bound and not potentially regulated. So hence, we wanted to ask whether P53 could regulate the expression of these targets, and we could see that it could in our model system. And I just, in the interest of time, I'm going to just say, I'm going to cut again to the chase here. We did some further investigation of where these were. These were in enhancers and promoters of these various genes, these binding sites. And I'm not going to talk about this, but here was a data in collaboration with Raul Rabadán, where we looked at just human tumor studies that are available online. Most of these are TCGA studies, the Cancer Genome Atlas studies, and we could see very strong correlations between many of these targets, including P10 and mutation of P53, meaning loss of expression being associated with a mutation of P53 for many of these targets in actual human samples and across many types of cancer. So I'm going to basically end by saying, I'm going to skip through these slides because this is just detailed evidence saying that the P10 site actually can act as a enhancer element for P10 expression. So a P53 binding site in the enhancer for P10 could actually regulate and also have some effect on the phenotype of the MCF10A cells. But anyway, so I'll just finish up in terms of this story just to say that, that we have under normal low-stress conditions, we have basal targets that are expressed by P53, and many of these are tumor suppressor genes, and that in combination between these basal targets and acute stress targets, these lead to tumor suppression, and that when mutation of P53 occurs, it's going to screw up the ability of the cell to regulate these basal targets and these stress response targets, which can lead to reduced expression of tumor suppressor genes and the stress response genes and spontaneous tumorigenesis. So, okay, so to summarize briefly, P10 is a tumor suppressor. It regulates a variety of pathways. It's regulated the transcript translation, post-translation, and protein interaction levels. P10 and other tumor suppressor genes are transcribed by P53 under low-stress conditions in the body, and mutation of P53 will lower expression of a dozen tumor suppressor genes, including haploinsufficient tumor suppressor genes. I mentioned P10, but there are other haploinsufficient tumor suppressor genes on the binding that are targets for P53, including P21, CDK1A, and STK11 and PHLDA3, which could trigger tumor initiation or progression. And so finally, I'd like to thank the people that did the work. For the Deepti Mather is the first author on the metabolic analysis pathway paper, and Curie Pappas is the first author on the P53 regulation of P10 and other tumor suppressor genes pathway. And we also couldn't have done it without a lot of different collaborators, including Emily Bernstein's group here, which was really instrumental, and also Jim Manfredi's group, who did a lot of the work with us in terms of P53 regulation. And and also some of our other collaborators that still at Columbia and other institutions. And thank you very much. Thank you.
First of all, congratulations on highlighting this factor. My question is about your work and human aging. I'm not talking about old people or old animals, but simply the biology of aging and the Hayflick work. Have you focused on the link of this factor, P10, and the biology of aging?
No, actually, I haven't, but there's some probably the best work in this area is in C. elegans, where age 1, which is the key, or one of the key genes that regulates aging in worms, is P10 kinase. So it's known that mutations that lead to reduced activation of the P10 kinase pathway lead to longer lifespan in that model system. And it's also known that when P10 is lost, it causes shorter lifespan. Basically, enhancing the metabolism of the animal leads to shorter lifespan. It's also known in fruit flies, not exactly the same genetics apply, but S6 kinase, for instance, a mutation downstream on the pathway, leads to enhanced lifespan in fruit flies that makes the animals much smaller. I will also add that a lot of the genetics on this pathway lead to a smaller animal size, which also could have the indirect effect of just having altered metabolism. So it's a little complicated, but overall, we also know that diet restriction in a variety of different animal models leads to increased lifespan. And since this pathway is so involved in regulation of glucose and other metabolic components, there's just, I think, a lot to learn here. The other area I think that is very important to touch on is the relationship between this pathway and obesity. We know that there's a lot of the signaling pathways. The insulin signaling pathway, is very active in adipose tissue. It's very active, obviously, in muscle. It's active in the liver, in endocrine signaling systems. And I think there's a lot left to learn there. For instance, to give you an example, we can see huge levels of PREX2 in adipose tissue, and we don't know why it's there and what it's exactly doing. It could be some very specific adipocyte regulatory signal that we just don't understand. So, and obviously, aging is related to, obviously, not only health, but also disease prevention. So this pathway cuts both ways in the sense that when you activate it, you reduce lifespan, and when you activate it, you also get various different disease manifestations. Excuse me. For another example would be insulin resistance, where it's pretty clear that a part of the problem here is this, with persistent insulin signaling, is in peripheral resistance in part, and even though that's not a really completely well understood area, I do think there's a lot more to learn with regard to how these components in this signaling pathway are affected in disease like diabetes.
Ramon, amazing body of work. You alluded to some of the challenges of targeting this very centrally important pathway toxicity. That combinations might be an approach.
Maybe not.
Or how do you see efforts to more specifically target this pathway as being a way forward? Is that going to be sufficient to
I
get
think
inhibitors that work?
one of the problems with drug development is extremely slow and tedious, and it's not, you make, literally, I think there's been like 5 or 6 generations of PI3 kinase inhibitors at this point. And what we find out is that the first generation actually, even though they inhibited PI3 kinase, they also inhibited mTOR, they also inhibited, believe it or not, BRD4, they bind to BRD4. And so they're extremely toxic and really polyvalent, meaning that they hit just a ton of different targets. And the more recent ones, they had on-target toxicity, meaning they could often lead to induce hypoglycemia rapidly, and which is a problem, right? But what's fascinating is some of the most recent drugs, which are very much more precise in inhibiting, let's say, AKT 1, 2, and 3, or in inhibiting PI3 kinase alpha, they are not seeing the same level of acute metabolic problems that they were seeing with the prior perfect group. The problem is every class of drugs that comes forward is like the best drug. So, but the prior version was just really fraught with problems, fraught with toxicity, some off target, some on target. Now it seems like this most recent group appears to be, so anyways, I'm hopeful that in combination therapy, there may be a window where some of these drugs could potentially work. But I'm always going to be glass half full about this. So, but it'll take time. Yeah.
One more question we've got.
So, is there data showing that giving the MDM2 inhibitors upregulates P10 activity and affects the downstream metabolic?
That's a really good question. We saw that only in one. I flashed through that slide. We tested 3 cell lines, and we found that the MCF7 line, which is the one with the very low levels of P53, could lead to like a 25% reduction in P10 when we treated. I'm a little worried about cancer cell lines in general because they can be so messed up. It's hard to know when you get a negative result why. But 2 of the lines didn't show a response, but they also had very strong binding to P10 already. So again, also really hard to follow. But my just a final point would be, we're starting collaboration with Ron Hoffman because he's doing some work with Nutlands, and he's seeing some activity in the clinical trial here. So we want to take a look and see if some of these targets are being regulated in that setting.
Okay. Let me thank you.